Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. We are from Group 11. For today's topic, we are going to present you about the role of biotechnology in protecting plants against weeds. Our group members are Alifiandra Soni Putra with student number 208, Abil Milzam Syati with student number 229, Raihan Akbar, which is myself, 210, Humam Yusuf Abdussalam with student number 215, and Dian Ardhana Yoga Pamungkas with student number 217.
And then, what is weed? A weed is a plant considered undesirable in a particular situation. We can make it simple: a plant in the wrong place. Weed is a plant that not valued where it is growing and is usually of vigorous growth. It prevents the plants that you want from growing properly, and we must remove them to make optimum yield. And what is biotechnology? Biotechnology is a technology that utilizes biological systems, living organisms or parts of these to develop or create different products. In this context, we use biotechnology to control the population of wheat. Biotechnology in agricultural means range of tools including traditional breeding techniques that alter living organisms or parts of organisms to make or modify products. Biotechnology improves plants or animals or develop microorganisms for specific agricultural uses. Modern biotechnology today includes the tools of genetic engineering. Biotechnology in weed control. Nowadays, biotechnology has provided new dimensions to herbicide technology. Transgene technology has generated herbicide-resistant crops, or HRCS, which have had a profound impact on the herbicide market. The same technology has the potential to make crops better competitors with weeds through improving competitive traits or making the crop more allelopathic. Living biocontrol agents can sometimes be applied to weeds much like a herbicide. However, biocontrol has not been widely applied in agronomic and horticultural crops for weed management because of a number of failings when compared with chemical herbicides. However, biotechnological advance may change the equation favoring biocontrol in some situations. But we have opportunities for the application of biotechnology. First is the development and use of bioherbicides, the discovery and use of naturally occurring herbicides such as fungi, bacteria, and virus as biological control agents. The second is the genetic improvement of crop tolerance to herbicide and the use of genetically engineered microorganisms for other areas of weed management such as the decontamination of herbicide spills or the protection of selected crops against herbicide injury.
Okay. Advantages of utilization of biological weed control agent. First, a high degree of specificity for the target weed. Second, no effect on non-target and beneficial plant. Third, absence of weed-resistant development. Fourth, absence of residue build-up in the environment. Fifth, potential impact from biotechnological research and development. Disadvantages of the production and use of bioherbicide. First, bioherbicide have to be registered with the Environmental Protection Agency, and the registration process may be lengthy. Second, suppression or killing of weed by bioherbicide may be a slow process. Third, stability of bioherbicide under field condition is highly dependent on environmental condition. Fourth, production of a bioherbicide for the large-scale application may be an expensive process. Fifth, numerous fungi need to be discovered and developed as bioherbicide.
My name is Humam. 134, 180, 250. Allopathy refers chemical released into the environment where it affects the development and growth of neighboring plants. Allopathy is derived from the Greek word allelon, means each other, and plantos means to suffer injurious effect of one upon another. Advantage of allelopathy: first, limit competition for space, light, nutrition, water. Second, autotoxicity, chemical produced inhibits germination of its own seed. Third, allopathic with suppression through the use of cover crops. Control with allopathy. Allopathy extracts have been successfully used for organic weed management. Allochemicals are diverse in nature and structure, and thus lack common mode of action. This biotoxic activity of allochemical is responsible of growth suppression of weeds. Allococcus, when one plant species release chemical compounds, either directly or indirectly through microbial decomposition of reduce that affect another plant species, including allopathic plant in a crop rotation has part of an intercropping system may provide a non-herbicide mechanism of weed control. Then allopathic compounds. Certain plants have allopathic abilities which are thought of affect germination, growth, and yield of other cultivar plants. These allopathic compounds consist of three main compounds, namely fixatin, isoflavonoid, texin, and glucosylflavonoid. All three are included the flavonoid group, which is group phenolic compounds. In addition, the production and development of chemical resistance. The chemical resistance is known as phenomenon which called allopathy. The compounds that cause allopathy are called allopathic substances.
So the next topic we're going to discuss is biocontrol of weeds using plant pathogens. A variety of chemical, mechanical, and cultural methods are usually used to control weeds, but in recent years there is an increasing attention for the use of insects, fungi, and other plant pathogens to control weeds biologically. So this method is called biological control or biocontrol. On the left we may see our dear little friends larvae of the Cactoblastis Cacrotum stricta moth, which fed on the common prickly pear cactus Opuntia stricta in Australia back in 1920s. At that time, biological control shows a promising way to control weeds. The common pathogens used for this method is fungi. Why? Because fungi have a high reproductive capacity, a short generation time, and are often able to survive as resting structures or as saprophytes during periods when host plants are not even available. Two basic approaches to control weeds using fungi are classical biocontrol and inundative biocontrol. Okay, let's get deeper to classical biocontrol first. Classical biocontrol is directed at long-term control of weeds and the permanency. So let's get deeper to classical biocontrol first. But classical biocontrol is directed at long-term control of weeds, and the permanency of the control is more important than the time required. But the things to be noted here: the pathogen must be host specific and self-sustaining. In other words, it should be able to survive and multiply after its release and in its new environment, and not attack non-target plants, in this case our crops. And we have the table here for as fungi is used in classical biocontrol with its host. And the next one we have inundative biocontrol. The pathogens are usually indigenous or naturalized, occurring naturally on weeds in areas when they need to be controlled, but not at levels sufficient enough to significantly affect the survival of weeds. The fungus is mass-produced and a large number of spores are sprayed onto the weeds in the same way as the chemical herbicides. For these reasons, inundative biocontrol agents are often referred to mycoherbicides or bioherbicides. Here we have 3 microherbicides which have been registered for use. They are Divine. Second product we have Collego, and then lastly Europe. In Europe we have Biocon.
My name is Abil Milzam, and I will explain about biotechnology in weed control. First, next, biological control method. Biological control methods for weeds usually cause less contamination of soil, water, and food with unwet synthetic compounds, and they do not contribute to soil erosion as does tillage, the main non-chemical method of weeds management. A less common strategy is the use of living organisms, including livestock, insects, nematodes, fungi, and bacteria to target weeds. Many biological agents target specific weed species, while livestock are relatively more generalist in wide consumption and may avoid eating certain weeds. Next, biotechnological approach in weed management. Through biotechnology, any biological system, living organisms are parts of this intelligence to develop or create different products for controlling weeds population. There are different approaches for biotechnology weeds management. First, development of herbicide-resistant crops. Second, improvement of biocontrol agents. Third, development of transgenic allelopathy in crops. Fourth, characterization of weeds using molecular systematic. Okay, next, development of herbicide-resistant crops. If we are able to develop herbicide-resistant in crop and apply total herbicide to the all plants in the field, will they accept herbicide-resistant crop through increased selectivity of the herbicide? Technique involved in developing herbicide-resistant crop. Herbicide-resistant crop can be developed through traditional plant breeding, and biotechnology include in vitro mutant selection at the cell or tissues level, somatic hybridization, microspore and seed mutagenesis, and plant transformation or transfer of clone genes into susceptible plants. Next, improvement of biocontrol agents. Any living organism or biological entity improvement for lowering the population of weeds is called biocontrol agent. For example, parasite, predator. Predator is insect mates. Pathogen is fungi, bacteria, virus. Next, other biotechnological agent. Biotechnological agents is crop or weeds. Okay, next, it will explained by my friend Yoga.
Characterization of weed using molecular systematic. Molecular systematic is the use of molecular genetics to study the evolution of relationship among individual and species. There are some advantages of molecular systematic. First, obtain phylogenetically informative character from any genome of the organism. Second, lesser chance of getting misinterpreted gene character. Third, applicable where resistant gene tests are difficult, complex, expensive, and unreliable. Fourth, applicable where pathogen is quarantined. And fifth, provide the decisive data for differentiating two similar species. So that presentation from our group. Thank you.